Welcome back to the course on fluorescence spectroscopy. This is part 3. We're going to talk about time-resolved fluorescence today. It's really one of my favorite topics, I think, because the time-resolved measurements can remove many of the ambiguities that show up with just steady-state measurements. So what are time-resolved measurements? In principle, it's simple. You have a pulse of light, it impinges on a sample, you collect the emission, and you see that the emission decays. And that decay is the inverse of the lifetime. Typically, that occurs on the nanosecond time scale for just about anything that you might be measuring. So let's look a little more closely at the decay law. We can write down a differential equation for the change in the number of molecules in the excited state versus time, which is simply it's being decreased by the radiative rate and the non-radiative rates. Solve the differential equation and you get a single exponential decay. What is the average lifetime? You can determine the average lifetime in two different ways. One way, if you're doing it graphically, and it was a very simple decay, it's the 1 over E position in time for the intensity decay. Or you can define it as an integral of the time times the intensity decay, and you can call that the average lifetime. We don't always have a single lifetime, so this is a very simple case. Let's look at a typical result from a time-resolved measurement. You might not understand every part of this slide right now, but you will by the end of this lecture. So I chose a very simple one, green fluorescent protein, which was, I think we mentioned that before. And there are 4 different lines on this graph that are important for understanding. The first is what's called the impulse response function. That's how the instrument responds to an instantaneous pulse of light. That's a rather complex area because it varies greatly between the types of detectors that you have, and they continue to evolve. But in every intensity decay, one of the first things you have to measure is the instrument's impulse response function. Then we have actually 2 lines here. They don't look like 2 because there's a perfect fit. The dots, the data points, that's the number of photons measured at each particular time in the decay. And there's a solid line going through there, which is the data fit. On the bottom, we have the residuals, and that's basically the difference between these 2 lines up here, between the calculated decay and the measured decay. And I'll mention this early. We're going to use a fit parameter, chi sub r squared, and a chi sub r squared around 1 means that basically the only noise, basically the model fits and the only deviations are due to the Poissonian noise and the number of photons that you count, which can't be avoided. I'd like to give you a couple of biochemical examples just to show the utility of the lifetime measurements. An example here shows that a protein conformational change can be detected by a change in the intensity decay. And as you might guess, moving the tryptophan into the protein for some reason causes quenching and a more rapid decay of the fluorescence. If the tryptophan pops out in the presence of calcium, you have a different decay. So for the same protein, you can have 2 very different decays. Another example is a protein with two tryptophanes. There's been a lot of work on tryptophan fluorescence, which we'll talk about in a future lecture. And very often the goal of the measurement is to try to recover the lifetimes of the two tryptophanes that are in the protein. And that's a very difficult task. In this case, it was simplified by using site-directed mutagenesis, making the proteins with only one of the two tryptophanes. And as you can see, one of the tryptophanes in this particular protein has a much longer lifetime than the other tryptophane. And in this case, the results are plotted as a lifetime distribution analysis, which we'll not really talk about right now. I have some equations put on these slides. I'm not going to talk about them very much. They're just for those people who really like the mathematics. So why do we like lifetimes? One of the reasons we might like lifetimes is that they are independent of fluorescence intensity. So let's do a thought experiment. We have a cuvette here that's clear, typical cuvette here. We have another sample holder filled with blood, and we try to measure the intensity in there. And of course, it's the one with blood, the intensity is greatly filtered. The one without blood, you get a higher intensity. So steady state measurements are very dependent on the conditions. Here we have the decay times. If we measure the decay times of the same fluorophore in these two samples, assuming it hasn't bound to something in the samples, the decay times would be exactly the same, independent of the intensity. And this is also going to be true in the frequency domain, which we'll talk about in a future lecture. So the independence of the lifetime from intensity has become very valuable in a field called fluorescence lifetime imaging microscopy. I'm guessing that right now, about half of the high-end laser scanning confocal microscopes have the film capabilities in them. But the basic idea is, instead of whatever probe you put in the sample, instead of looking at the intensity of that probe in the sample, you measure the lifetime of the probe in the sample. In this case, we chose a chloride sensitive probe, and these measurements based on color, red, it turns out to code for a shorter lifetime. These are of course artificial codes, but you can do a lifetime imaging experiment, and from the color you can determine the chloride concentration at any position in the sample. So FLIM is very popular, and I'm proud to say I coined that acronym. The problem with time-resolved measurements is that there is no instrument available at present that can collect the entire time-resolved decay on the nanosecond timescale. So you can't take a sample, put a pulse into it, and just measure the decay at all the points you need to recover all of the information. Maybe they can do that at CERN, but there's no commercial instrument to do this. There have been attempts to make such commercial instruments, and I guess I'm telling you, Jeff, this just to understand why lifetime measurements have been difficult. There have been attempts to record the entire decay directly. There have been attempts to use a sampling oscilloscope to measure the decays, and these have just not worked well enough. There are always problems, seem to be due to light interference electronic interference with the electronics in any way. The companies who are trying to do that have gone out of business. So these direct measurement methods have been rejected in place, and instead we're using what's called time correlated single photon counting. So that's a long word. What does it mean? Well, one of the first things you have to remember is that during a fluorescence decay, all of the photons are not emitting at the lifetime. Okay, there's some of the molecules decay fast, some decay slower. It's like a radioactive decay. Some of the atomic nuclei decay faster, but it's always at a random point during the decay. And in order to do time correlated single photon counting, if you do it one time, you only get one photon, so that doesn't tell you much. You have to use a string of laser pulses, because you have to do this experiment many thousands of times. And what you do is you send in the pulse of light and you measure where you see the photon come out. Then you send in another pulse of light, measure where the photon comes out. And eventually you build up a histogram. And that histogram is the number of photons at any given time. And that histogram can be used to analyze and tell you the lifetime. There's a couple problems with this. One is that the instruments normally don't have delta function excitation. You have some type of excitation pulse, which can be anywhere from a couple nanoseconds wide to 100 picoseconds wide. But in any event, the sample is being exposed at different times with different intensities. And at each one of these positions along the intensity pulse, you're starting another time-dependent decay. So what you're measuring after you do this a couple thousand times is all of the decays that have happened due to excitation at all of these different points in time. That's called a convolution. The nomenclature doesn't matter very much, but the intensity decay that you measure is not really the actual intensity decay. This is the one at one point. It's actually the intensity decay exciting the sample at all these different time points. Let's say it's a nanosecond wide. You measure it at 10 or 15 different places. So your intensity decay is actually a sum of intensity decays with amplitudes proportional to the intensity of the light at that point in time. So that's why we have this convolution problem. Now, what happens in a instrument for time correlated single photon counting is simply that they have the electronics that measure the time from when the light pulse arrived to where the first photon was detected. And that's what all of this stuff is. I think it's a byproduct of probably defense research early on, but it used to be pretty complicated stuff, but it's not that complicated anymore. So that's how you build up this histogram from these photons that are emitting at different places. And I think it's this, another thing that these instruments have is threshold detection. And the pulses from a single photon are not always the same, but in typically there's noise pulses, and you set your electronics so you don't count most of the noise pulses. And I think this is the reason why the direct recording instruments have not worked very well, is they had no way of rejecting the noise. And another thing, the instruments are very clever, that even if the pulse height of a single photon varies, the electronics will measure the center of that pulse and will disregard its amplitude. But there is an expense you have to pay for doing single photon counting, and that is it requires a very low probability of detecting a photon per each pulse. Think about it. You have an excitation pulse here, and then you detect the first photon. The electronics is not capable of seeing the second photon. It's busy, and so it rejects that for a certain period of time. And if you had a really bright pulse and you had, maybe thousands of photons, you're only going to see the first photon, and you'll never see the rest of them. So the only way this works is if you have your system set up so you only count about 1 photon per 100 laser pulses. And if you do that, you can get the histogram that I showed on the previous slides, but you won't get that histogram if you have multiple pulses. Now, I suspect in the future we'll see instruments that can detect 2 or 3 photons per pulse, but for the time being, this is what you have to live with. This used to be a big problem. It's not a big problem anymore. And I think by explaining a little bit of the history, it'll help you understand how time-resolved measurements work. First thing that came out were nanosecond flash lamps in the 1980s. There were custom companies that would sell time-resolved instruments with them. They had maybe flash rates of 20 kilohertz, collecting only 1%. You have to set it up to get 1 photon per pulse. So it gives you 200 photons per second. That's not enough. And anyway, the sparks were about the spark you would get if you had an electric charge from walking around on a dry rug at home and touching it. And so you were almost lucky if you got 1 pulse. And then by the time you got enough data, the pulse had moved around, which means your impulse response function would have changed. And you notice it's quite wide here, which is typical of flash lamps. I don't know the exact width, but I'm going to guess it's 20 nanoseconds wide or something like that. Now, I'm not going to explain everything about the history of how measurements were, how lasers contributed to time-resolved measurements, except to say that it was as expensive as anything and as time-consuming as anything. These are basically large ion lasers that would pump dye lasers that would then have to be frequency doubled. To get this system to work, you'd probably have to have about 20 mirrors in perfect alignment, while at the same time having just the right path length that the photons are moving around, at the right speed so they bunch up, because they have to be mode locked by some feature on the end of the laser. And when you got done with this, you would wind up with long wavelength light, typically, which wasn't very useful, like rotamy, like 600 nanometers, not that many probes out there, and you would wind up with 80 megahertz light. And that was too fast for many measurements, because at 80 megahertz, it's about 12 nanoseconds between the pulses, and you need a longer time than that to let the entire intensity decay before the next pulse. So another complication, you would have to use frequency doubling to get a shorter wavelength. And you would also need a cavity dumper to decrease the repetition rate. So what the joke used to be, if you were reviewing someone's NIH grant and they wanted a system like this and he was a competitor, first thing you do is you give them the money to buy the system, because you know they're going to lose about a year of their life getting everything working. This got a little simpler with Thai sapphire lasers, but they still had many of the same problems. But what you really needed was a set of pulses where you had a good short pulse, and you could adjust the time between them, and that's what cavity dumping would do. It would take about one out of about 50 pulses and dump it and send it towards the sample. But that was all too expensive. What has really made the time resolved measurements much more friendly is they can now be done with pulse laser diodes, and you can't imagine how much simpler this makes the measurements. These large frame lasers would take something like 40 kilowatts out of the wall from 480-volt lines. It was just terrible. But now you can use pulse laser diodes. Not every wavelength is available, but enough are available. And the pulse rate is driven. You can decide what pulse rate you want, so you don't need a cavity dumper anymore. You can just say, I want this repetition rate, and you use the right laser head, and you can go ahead and do your measurements. So now it's completely, basically you can really have a lifetime instrument in a PC. It takes one board, and if you're not really interested in a lot of spectroscopy, there are simple instruments that are filter fluorometers. Put them on your desktop, and you can get time resolved decays, very good time resolved decays. I'd just like to mention the future. I don't know if this is ever going to become inexpensive enough or not, but there is a light source now called the supercontinuum lasers that have intense light at almost all wavelengths. The difficulty with them is you have to be able to extract just the wavelength you want. And as we mentioned with arc lamps, you could always get stray lights sneaking through, so you need very careful filtering and diffraction. But we have one of these, and it's quite convenient. So in order to, since the measured decay is a convolution with the excitation pulse, we have to do an analysis to get the true intensity decay, like the one on the first slide, the single exponential decay, because the measured data is going to be this hodgepodge of all of these other decays that have been summed up. So we need deconvolution software to extract the actual intensity decay. I just wanted to mention places where you need it and you don't need it. Mathematically, that's the symbol for convolution, but here's a measurement of the decay of a very short lifetime molecule. And as you can see, the instrument response function has a bump in it, and the fluorescence has a bump in it. And believe it or not, when you analyze this and you do the deconvolution analysis, it's a single exponential decay. Of course, the data doesn't look single exponential because of the bad instrument response function. If your lifetime is very long, you can ignore that part of the decay. Typically, for most of the measurements you're going to be doing here, you're going to have to be doing a deconvolution analysis, because the impulse response function of most instruments you use are going to be around two nanoseconds, and all these new high extinction coefficient dyes have very short lifetimes. So deconvolution will almost always be necessary to extract the decays. So back to the green fluorescent protein, the straight line on the log scale meant it was a single exponential decay. This is, and let's look at this impulse response function here. This is pretty good. Some instruments are much worse than that. And as you can see, the deviations, you have a chi-squared around 1, which means that the only difference between a calculated curve and the measured curve is unavoidable Poissonian noise in the number of photons that you've counted. And that's actually simple to calculate. Let's say you have 10,000 counts in a particular channel. The amount of noise in that is the square root of the number of counts. So the noise in that measurement is 100 counts. So what do you do when you do a time domain analysis? I like this chart. I've used it a few times. And if you're really mathematically inclined, on the next slide, I have the same chart with the equations on it. But what basically you're doing is you're doing your measurements, which gives you these, and it gives you your observable quantities. Then you use some assumptions for what the decay looks like, which is typically a multi-exponential decay, and you then do some type of analysis to fit them. The preferred analysis is nonlinear least squares. There's other ways of doing the analysis, but I don't think anything is better than the nonlinear least squares. And this is the slide I promised with equations for anyone who are feeling a little more geeky than I am at the moment. So let's look at a couple of measurements. This is an instrument with a terrible impulse response function. This particular phototube was actually designed to have a very good impulse response function, but you see it has a lot of ringing in it or something. This is a product of the phototube, not the lamp. And here we have a mixture of two fluorophores, one with a lifetime of 1 nanosecond, the other with a lifetime of 6 nanoseconds. The way you do the analysis is first you try with a single exponential decay. And when you look at that, you can just see that the data and the calculated decay don't match anymore. And that shows up by the deviations on the bottom. So since the deviations are not random, you know it's not a good fit. And if you look at the chi-squared, my old eyes can't read that at this moment, but it's a couple hundred. And obviously you reject that fit. If you now give the computer, say, oh, you can use 2 lifetimes, where you find everything turns out perfectly, and the data and the fit work out, no time-dependent deviations, and the chi-squared is around 1. So that's pretty easy to do, 1 in 6 nanoseconds. And the reason I'm telling you this is because closely spaced exponentials are very difficult to resolve. I think the mathematicians have some theory on that. So let's look at 2 more closely spaced decay times and see what decision you would make. So we have now 4 nanoseconds and 6 nanoseconds. We do a single exponential decay on that. Well, it's not bad. Deviations are relatively small. Chi-squared is near 3. Any simple experimental error could give you a chi-squared of 3. So you might mistake this for a single exponential decay. You do a double exponential decay, the fit improves. Most people would say, okay, this sample has 2 different decay times in it. The chi-squared is about 1. The point about this is that's all you can say. You can say the data is consistent with a double exponential decay. You cannot say it's not a triple exponential decay, because it will also be consistent with the triple exponential decay or more. So it's you might exclude this one on the basis of chi-squared, except this one on the basis of chi-squared, but you can't say there's not 3 there, but you can say there are 2 lifetimes there. Oh, and I just thought I'd show you this. I don't know why I thought you would find this interesting. But there's some really promising things coming out in the future for detectors. These are some of the latest detectors for Hamamatsu, uh, and they're called hybrid photomultiplier tubes. And the important thing about them, as opposed to that big lumpy decay I had due to the ringing in the phototube, these have what are called transitide spread around 30 picoseconds. They don't look like they're going to be expensive to make. So I think the time resolved instrumentation is going to get even simpler to use. What are the meanings of the term in the intensity decays? Because whenever we recover decay, it's a pre-exponential factor and a decay time, and another pre-exponential factor and a decay time. This is a 2 exponential decay. So if you looked at these two numbers, you might say, oh, there's a lot more of the 5 nanosecond component because the pre-exponential factor is four times larger than the 20 nanosecond component. That is a misinterpretation of the data. The actual contribution of a component to the steady state spectrum is the area under the curve. It's the alpha tau product. So for this particular intensity decay, artificial one, the 5 nanosecond and the 20 nanosecond decay times have the same total contribution to the fluorescence. The only thing that's different is lifetimes are different, and that change, but the area under the curves is the same. What are the limits of resolving multi-exponential decays? So we set out to do this one time, and it was very instructive. We wanted to see if we could resolve 3 closely spaced fluorophores in lifetime. The reason we did this is because if you look at the literature on protein fluorescence, where you'll almost always find 3 lifetimes for the protein spaced by about a factor of 2 each, and that's taken as the right answer, and I think in some cases it's just an illusion. These lifetimes are a little longer, but you can see each of these 3 fluorophores is a single exponential decay. So now we mix them together and asked what happened. And we mix them together, and it doesn't look too bad. It looks sort of single exponential, but the chi-squared is rather high. So you'll say, no, it has to be more complex than that. You go and analyze it with 2 lifetimes, and you get a quite reasonable chi-squared. And then you get 3 lifetimes, and you get about a perfect chi-squared. But in the real world, you wouldn't know which of these to accept, whether it's 2 or 3 components, because these are just too close together. So I regard this sort of the limit of what you can resolve with time-correlated time-correlated single photon counting. That is 3 lifetimes by about a factor of 2 in lifetimes each apart. That's about as good as the resolution gets. Jim Demas down at the University of Virginia did this also numerically in terms of just showing how hard it is to resolve multi-exponential decays. He created 2 decays with very different pre-exponential factors and different lifetimes, and he found them to be virtually indistinguishable. This is called a correlation between the parameters. In any multi-exponential decay, the alphas and the taus are highly correlated, and you can change one and compensate for that one by changing the other, and that's what's made the analysis difficult. There is a way to increase the resolution of multi-exponential decays, and this is your introduction to global analysis. And global analysis is very powerful. What it is basically, in the case of this mixture, it says instead of measuring the intensity decay at one place, we're going to measure the intensity decay at different wavelengths. So now we have multiple data sets for this one mixture. Well, I just happened to put the single data set results here, and as you can see, that they can be resolved. But there's a thing you can do with the multi-exponential decay. You can say, okay, I believe that the compound, there's two compounds there and they have the same lifetime. You don't know what that lifetime is, but you think they're the same. So you do an analysis of all of those decays together, forcing it to have only two lifetimes for the whole set of data, and you wind up with something like this. These are the single wavelength measurements, and you can see that the lifetimes vary somewhat. These little red things in the middle are the uncertainties from the least squared analysis. In general, it seems to me that the least squared analysis will always underestimate what your actual uncertainties are. So, whatever the computer tells you the uncertainties are, you can multiply them by a factor of 3 because the least squares analysis will underestimate them. But this uncertainty is broken down when you do the global analysis, because here's one of the single file fits, and here's a fit that we did that it said, well, there has to be two lifetimes, but the amplitudes can be anything, and as you can tell, it greatly improves your measurements. Oh, I forgot to make an important point. We call these the chi-squared surfaces, and I think this is the best way to know how good your parameter values are, because the way the chi-squared surfaces calc is done is you have your final result, you change one of the result values by a little bit, refit the curve, and get the result, see what the chi-squared is, and you do that 20, 50 times across the sample, and the chi-squared surface is, to my mind, the absolute best way to know the uncertainty in your measurements, because you can do the statistics or what the F statistics are, and they can tell you how much the chi-square can increase before you have to say, no, the data are inconsistent with that. So anyway, all right, and I think, yes, so now we're coming to the end of this talk that's longer than all of the others. And the question, last thing when it closes, what are the origins of multi-exponential decays? We've been talking about them. It can be multiple chromophores. The proteins can have multiple tryptophanes. You can have probes in different environments, say some partitioned into a membrane and others not partitioned to the membrane. Different conformations in the proteins. Rotamers, the fluorophores, in a different contribution. Later on in these talks, when we talk about energy transfer, where there can be a distribution of distances between the donor and the acceptor. That will give you another form of decay, which we haven't considered. There's in quenching, there's other types of decay laws. Quenching has a decay law with a square root of T in it. Other decay laws are lifetime distributions. The most common cause for a multi-exponential decay, in my opinion, are either impurities in the sample, scattered light, or systematic errors in the measurements. So before you go crazy looking for some small component, make sure your data is good. And I guess one of the things I always think is if it's a very minor component in the decay, you know, ignore it, go do your next experiment, because if it's real, it's going to show up the next time rather than chasing after it. Well, anyway, that's the end of part 3, and I thank you very much for your attention, and we'll see you next time.